I decided to give a little bit of a hybrid talk, talking both about cichlid fish and about butterflies. So I work on different study systems, but the questions are all kind of in the realm of what facilitates rapid speciation. And one of the aspects I'm particularly interested in is how hybridization or admixture, so the mixing of genetic variation between species, can facilitate speciation. In general, my work has been driven by the curiosity about why are there some lineages, like for instance, the peacock spiders, which I also study, are able to speciate extremely rapidly, whereas their close relatives in the exact same place in Australia do not speciate in the same time frame. So why is it that biodiversity is so unevenly distributed across the tree of life? And one of the things that generates very rapid, or one of the processes, I should say, that generates very many species very rapidly is adaptive radiation, which most likely all of you know, but just to recap, which is when a species rapidly diversifies into multiple species, utilizing different ecological niches. Like for instance, the famous Darwin's finches that have diversified on the Galapagos Islands and are adapted to different seeds that these species are specialized on. So adaptive radiation requires ecological opportunity. Without those islands, without empty niches on those islands, they couldn't have diversified. And it also requires the evolution of some reproductive isolation barriers that allows them to adapt to different niches without merging back into one species all the time. So there needs to be some kind of assortative mating, for instance, evolving that allows them to mate mostly within their own species. And both of these require genetic diversity. Ecological opportunity is no good if the species cannot adapt to the ecological opportunities, and that requires genetic diversity. And also reproductive isolation barriers, such as, for instance, assortative mating, requires the genetic variants that underlie, for instance, mate choice and preference for different male color phenotypes, for instance, and also the genetic basis of the male color type needs to be variation in those so that the species can actually diversify. So genetic diversity underlies kind of the base of adaptive radiation. And that's also something that will come over and over in my talk. One of the things I'm particularly excited about is that some lineages seem to be particularly prone to adaptive radiation and diversify over and over again. Like for instance, the Anolis lizards have diversified into different ecomorphs like the brown crown giant and the grass bush and the trunk ecomorphs on multiple islands on the Caribbean, or the tetragnatha spiders on Hawaiian Islands. Also on each island, there's multiple different ecomorphs, so different species adapted to different ecological niches. And it's kind of the same set of ecological niches on the different islands. And these species have diversified into similar sets of ecomorphs on each of these islands. And another classic example, which I've worked on, are the cichlid fishes, where we have these major lakes, Tanganyika, Victoria, Malawi. And in each of these lakes, we have these amazing adaptive radiations of cichlids that have evolved in these lakes. So we were looking at why do some of the lineages, like the cichlids you can see in these pictures, diversify very, very massively, whereas other cichlids actually don't diversify. So cichlids is a very, very big group of over 1,000, like 1,700 species that are currently described, but there's probably way more than that. And most of them are concentrated in few radiations, and many of the lineages have actually not diversified, they just contain 1 or 2 species. So we looked into what are the factors that predict if a species radiates and diversifies into many species or not. And with that, together with Matt McKee, who is now a professor at Monash University, we were generating a phylogeny of all described species of cichlids, and then mapped across this phylogeny what kind of predictors might be associated with the speciation rates. So some lineages like Lake Malawi or Lake Victoria cichlids have very, very high speciation rates, and these phylogenies allow us to infer these speciation rates. And we can then look, are those lineages that diversify rapidly, for instance, more often found in deserts or in high rainfall areas. And what we find is that there's some predictors which are consistent with previous results using different methods that are very interesting, like for instance, a large depth gradient. A large depth gradient, so deep lakes, provides more ecological niches and thus more ecological opportunities than, for instance, a shallow river, which is generally quite poor in ecological niches that these cichlids have access to. And another very important predictor is a lineage-specific one, which is species that have strong male ornaments are more likely to diversify. So basically, again, what I said before, ecological opportunity and some kind of reproductive isolation, which in these cichlids is mostly assortative mating via male coloration, that predicts which of those lineages diversify. And yet there are some parts of the tree which we just really cannot explain with any of the factors we looked into. And one of the examples is the Lake Victoria cichlids. This is a very interesting radiation, but it's only one lineage that actually diversified. There are 5 lineages that colonized the lake. 4 of them did not diversify at all, and the 5th diversified into 500 species. And they are all in the same lake. So you think they have the same ecological opportunity, and yet there's this massive difference in speciation rates. It could well be that this one was just there first and diversified, and then when these ones came later, the ecological niches were already used up. But that's not what we find. Looking at sediment cores, Moritz Muschig in the group of Ole Seehausen was looking at teeth in the sediment cores. And what he found is that right at the beginning of the formation of the lake, there are lots of teeth that are clearly assignable to this radiating lineage, but there's also teeth of these non-radiating lineages of those that have colonized the lake but did not diversify in the lake. And they're also there right at the onset of the lake. So they were there basically at the same time. So there's something special about this one lineage which can diversify much faster than these other cichlid lineages. And that's something that I wanted to explore during my PhD, and I'm still working on until now. So basically, this Lake Victoria region, it's not just Lake Victoria, it's also other lakes in the region, so all north of Tanganyika and Malawi. There's multiple lakes, each hosts a community of cichlids. Here you can see about 500 for Lake Victoria, about 50 for Kyoga, 10 Albert, 60 Edward, and altogether there's about 700 species. And the same lineage which diversified in Lake Victoria is also the one that diversified in all of these other lakes. So there's really something very special about this one lineage that seems to diversify over and over again in each of these lakes in the region. So I wanted to ask what makes this one lineage of cichlids so radiation prone. So for that, we sequenced cichlids representatives of all major drainage systems in the region that are or have been in contact with the region in the past. And with that, so we used rat sequencing, so we generated a giant tree. And because it's really hard to read anything, let's just zoom into this region here, which contains what we call the Lake Victoria Region Superflock. So that's the 700 species from the Lake Victoria region that form part of these radiations. And then the closest relatives I found here are cichlids from the Congo drainage system. And the next closest relatives are cichlids from east of Lake Victoria region and from the Upper Nile, which is actually part of the Lake Victoria region. And so I wanted to know, where did these cichlids come from? What are the closest relatives? And one could conclude, looking at this tree, that perhaps the Congolese lineages are the closest relatives, and perhaps the cichlids in the Lake Victoria region colonized from the Congo and then radiated. But when I tested if this tree is actually a good representation of the evolutionary history, or if there's any evidence of hybridization, I found very strong evidence of contributions from the Upper Nile lineage into the base of all of these radiations. So all radiations show the same evidence of a hybrid origin between the Upper Nile lineage, where we have 2 representatives, and the Congolese lineages, where we also have 2 different species from. So with lots of different tests of hybridization and generating dated phylogenies, I inferred this scenario, where about 20% of the ancestry of this Lake Victoria Region Superflock is derived from the Upper Nile lineage and 80% from the Congolese lineage. And these 2 lineages are, with a quite large confidence interval, but these 2 lineages are quite divergent. And then they likely hybridized around 150,000 years ago. And these dates fit quite nicely. Again, also the paleogeographic context has quite large confidence intervals, but it fits more or less the knowledge we have about how the drainage systems were connected or disconnected. So about 4 million years ago, when the Upper Nile and the Congolese lineage were not yet split, there was basically like one drainage system in this region of the Lake Victoria region. And then about 2 million years ago, there was uplifting of this region that separated what later became Nile drainage system from what later became the Congo drainage system. And so this fits more or less when these 2 lineages might have split. And then in the time around 150 to 120,000 years ago, there were lots of periods of very high humidity. And probably in one of those, perhaps via the Malagarasi, there might have been another connection where the Congo lineage could have come back into this Upper Nile drainage system. And this is probably when the lineages might have come together and hybridized. And nowadays, the lakes are mostly separate. There's well, in this case, for instance, between Lake Edward and Kivu, there's volcanoes that separate them, so they're totally separated. And in other cases, it's mostly rapids and waterfalls that prevent cichlids from moving between lakes. So in short, what we found is that the Nile and the Congolese lineage came together in this region, hybridized, and of this hybrid swarm of this mixed population, all of these radiations evolved. So if this hybridization really facilitated the diversification, we would perhaps expect something like this, where the Congolese ancestor and the Nilotic ancestor together gave rise to a diverse hybrid swarm, combining the genetic variation of those 2 lineages. And then, can't see my cursor anymore there. And the different species might then have arisen via sorting of these different Congolese and Nilotic alleles into different species that are different mosaics of these ancestral lineages. And by looking at the genetic variants that show evidence of divergent selection between the species in Lake Victoria, this is more or less what we find. So we find that there's many sites which are shown here where some species have fixed the Congolese allele and other species have fixed the Upper Nile allele. So like for instance here, this would be a site where these 3 species have fixed the Upper Nile allele and these 2 species have fixed the Congolese allele. This one has again fixed the Upper Nile allele. And so across these sites, you can see that the different species are different mosaics of these 2 lineages. But this is only true for genomic regions we found to be under strong diversifying selection. Overall, genome-wide, they all share very, very similar ancestry signatures of both of these lineages. And it's quite highly correlated, or some regions are mostly of Congolese ancestry in all species across all radiations, and other regions are mostly of Upper Nile lineage ancestry in all radiations, which really suggests that it was a single hybridization event at the onset before this lineage diversified into lots of species. And for some of these sites, we actually know what they do. And one of them is the long-wave sensitive opsin gene, which is important for seeing red coloration, which is very important in Lake Victoria because the light regime becomes much more red-shifted towards deeper water. And what we see if we generate the tree at this region is that there's two deeply divergent haplotype classes, one of which is associated with clear shallow water cichlids like algivores, and the other one is associated with turbid deep water cichlids like these detritivores. And when we look at where the Congolese and Upper Nile lineage haplotypes go in this tree, we see that it looks like the haplotype class 1 is derived from the Congolese lineage, and the haplotype class 2 is derived from the Upper Nile lineage. This suggests that the combination of these two lineages at this ecologically relevant gene facilitated speciation by providing the genetic variation that allows them to adapt to different water depths. And so this could really have facilitated and sped up rapid speciation, because when you think of new mutations, most of them are deleterious. It takes very long to change via new mutations, whereas if there's standing genetic variation, it can be much faster. So new species can evolve by selection on pre-existing genetic variation, which is much faster than long waiting times for new mutations. But particularly at ecologically relevant genes, if they get used over and over again in speciation events, which is what we see in the cichlids, the genetic variation gets depleted by repeated sweeps, selective sweeps. And so via just standing variation, the many successive speciation events, like we see in Lake Victoria, for instance, where 500 species in a very short period, standing variation may also not be providing enough variation that allows them to diversify as fast as they did. Whereas with admixture, if two genetic lineages that have very different genetic variants come together, combine their genetic variants, very large standing genetic variation is produced, which can then be selected into lots of different species. And this is what we think happened in the Lake Victoria cichlids and what our data support. So to get back to our question, what makes this one lineage of cichlids so radiation prone? Basically, it evolved from hybrid ancestry and thus started with very high genetic variation that could be selected into different species. And remember, it's just 150,000 years ago that all of these cichlids evolved. So it's extremely fast speciation. But it's actually even faster than that because Lake Victoria was completely dry for 4,000 years and only refilled 15,000 years ago. So all of these 500 species that we think now are found in Lake Victoria, and this amazing diversity that we see in the lake, like when you go there, it's just full of cichlids, and every ecological niche you can imagine has a cichlid adapted to it. We wanted to know if this all really evolved in just 15,000 years. So did the 500 species really evolve in just 15,000 years since refilling of the lake? For that, we now generated whole genome resequencing data. So from Lake Victoria, we sequenced over 300 specimens, usually like up to 3 per species, and then also representatives of all major ecological guilds of all the other lakes. And particularly noteworthy are Lake Edward and Lake Kivu, which are deep rift lakes and therefore may not have desiccated during this dry period. And the Kagera lakes we thought might be important because they're higher up in a more humid climate, so perhaps they also at least contain some swamps, which might have been important. So we wanted to know how are all those cichlids related with each other, and how did the much younger Lake Victoria radiation, did it get assembled from different lineages that colonized the lake independently? For instance, fish eaters and algae eaters and insect eaters, did they colonize independently, or did they really evolve within the lake? So if you have a look at the genomic structure within the lake, this is what we see. If we make a PCA of, in this case, 293 genomes, we see that they mostly cluster by ecology. So here the different symbols and colors are showing us the different ecology. So here we have multiple species of egg and fry eaters that all go together. All the different predator species go together. And here's a clade of detritivores. So we see that the ecologically similar species are also genomically similar and perhaps closely related to each other. So we wanted to know if, for instance, the piscivores, the predators that we see to cluster together in Lake Victoria, if they cluster together because they have independently colonized the lake, or if they did evolve once in the lake and then just diversified into a predator clade. So for that, we need to now add in the other cichlids from the other lakes. And we see that the Kagera Lake cichlid, so Kagera is over here, these more humid lakes at higher altitude, they go very, very close to the Lake Victoria and Lake Kyoga cichlids. So Lake Kyoga is basically just downstream of Lake Victoria, basically an extension of Lake Victoria. These two lakes are still connected nowadays. And the other lakes, which here I call Western lakes, which are in the west of Lake Victoria, they are all separate from the Victoria radiation. And so, for instance, our fish eaters from Lake Edward and Kiowa and Albert do not go together with fish eaters from Lake Victoria, but instead the fish eaters from Lake Victoria go together with algae eaters or insect eaters or egg and fry eaters from Lake Victoria. So genomically, the Lake Victoria cichlids are all more similar to each other than to any of these other radiation members, suggesting that they might have evolved within the lake. So that's very cool. We then also looked at the phylogeny of the Lake Victoria Region Superflock and the cichlids that are closely related. But what we see here is that the Lake Victoria radiation, which is this group here, all really forms nicely one phylogeny, one monophyletic clade. And the closest relatives are cichlids that are found just outside of Lake Victoria, the southern generalists. We caught them in a stream, in a little stream and swampy area just south of Lake Victoria. And the northern generalists, which is the next closest relative, basically the sister group in this tree to the Victoria radiation, they are found actually in Lake Kyoga, are generalists in Lake Kyoga that form the sister group to the Victoria radiation. So one could say that, okay, these two generalistic lineages are the closest relatives, and perhaps this radiation evolved from these generalistic lineages. But as I told you before, a tree is often not a very good representation if you have hybridization, and like the 150,000-year-old hybrid origin of the entire Lake Victoria Region Superflock, where the Congolese and the Upper Nile parental lineage came together and hybridized. We actually find evidence for something similar happening again at the origin of the Lake Victoria radiation. So I can't go into details of all of the different lines of evidence we have, but I just want to highlight two of them, which is that mitochondrially, the northern generalists, which are very similar, very close related in the nuclear phylogeny, show very, very deep divergence with the Victoria radiation cichlids. So mitochondrially, they're very distant, but in the nuclear phylogeny, they're very, very close. And we also find when we compare the southern generalists versus the Victoria radiation, we find that all different Victoria radiation clades, here shown as different dots, show high evidence of excess allele sharing with the Western Lake cichlid, so with these other radiations like Lake Edward and Lake Kivu. So this is a D-statistic, which is a test that tests for if both of these lineages here share similar amounts of alleles with the Western lakes, or if one of them shares more alleles with the Western lakes. And the statistic will be 0 in the absence of hybridization and becomes positive if the Victoria radiation has higher allele sharing with any of these Western lake groups that are shown here than the southern generalist does. So in short, what we find is that the Victoria radiation has more allele sharing with particularly Lake Edward and Lake Kivu cichlids than these generalistic cichlids that form the sister groups of the Victoria radiation. So suggesting that there was some additional contribution from Lake Edward Kivu relatives into the Victoria radiation. The fact that all Victoria radiation members show very, very similar these statistics suggests that this was an ancestral contribution and not into particular lineages. So it's not like, for instance, piscivores share more with the Western lakes than algivores do. They are all very, very similar, suggesting that it's ancestral to the radiation. And so basically, again, we conclude that this tree is not a very accurate representation of the evolutionary history, but instead it seems that there was an additional contribution from Edward Kivu cichlid lineage into the base of the Lake Victoria radiation. And we have evidence that both southern and northern generalists have contributed to the radiation. And so basically we have a second hybrid origin about less than 16,000 years ago at the base of the Victoria radiation. So basically, as I've shown you before, admixture variation can facilitate speciation, but then when those lineages that evolved from this admixture event, which is like the Congo-Nilotic admixture here, giving rise to different radiations with different radiation members, if they then come together again, they can radiate again as they each have fixed different genetic variants that in combination can again give rise to new species. Or shown in a different way, what we find is repeated cycles of admixture and diversification with at the origin of the entire Lake Victoria region superflock, so these 700 species of all the different lakes together. We have the Upper Nile and the Congolese lineage coming together. And then again, about less than 16,000 years ago, again, multiple lineages coming together in the Lake Victoria and radiating into many different species. And even within the Victoria radiation, we actually find that there's some clades that show very strong evidence of a hybrid origin, like for instance, these pelagic dwarf predators, which are little fish that are found in the pelagic and eat little other fish and copepods. And they kind of look like shrunken large predators in the shape and habitat of pelagic zooplanktivores. And genomically, that's exactly what they look like too. So genomically, they are 66% large predator ancestry and 34% pelagic zooplanktivore lineage ancestry. And while sadly, we don't know exactly which alleles are important for these different phenotypes, that would be very, very interesting to look at. We haven't done this yet, but we see that genome-wide, they are a mix between large predator and pelagic zooplanktivores, which probably explains their trait combinations that make them kind of intermediate between the two. And so we basically have another level of admixture where we have lots of cases of admixture giving rise to new ecomorphs like the pelagic dwarf predators within the radiation. So kind of 3 levels of admixture. And what we think is really important also to keep in mind is that why we think that these cichlids have diversified so rapidly and can do so over and over again, because they are able to still hybridize, and they thus have like super high genetic variation as a whole. And often conservation strategies are focused on specific species, but what really allowed these cichlids to bounce back after the lake desiccation is the fact that there were many lineages that came together. And so like preserving specific species may not be enough for rapid speciation and rapid adaptation. And it might be needed to preserve entire species complexes. And so in a different way, one could summarize it like this. Basically, throw a slow evolution through new mutations would look like this, whereas hybridization, evolution, or diversification through hybridization would look like this, where basically it's like combining two very different objects, mixing them up, and then lots of new species can evolve very rapidly. And so I was extremely excited about all these findings and was interested in finding out how general this pattern is. Are cichlids just weird, and is this just something cichlid specific, or is this a general pattern? And particularly also in the cichlids, we don't know that much about the genetic basis of particular traits. So we don't really know what are these specific Lego bricks in most cases. So when I moved to Cambridge, I wanted to switch study system to both figure out how generalizable this is and also to work on a study system where getting at the genetic basis of specific traits is a bit easier. So I went to Ecuador, and I have since been working with Heliconius and Ithomini butterflies. Here an example of Heliconius butterflies. And in a first project, I was working mostly on Heliconius together with Chris Jiggins. This is a very interesting group of butterflies because the different species show very strong Müllerian mimicry. So this is one species here, Heliconius erato, and Heliconius melpomene is another species. And wherever they co-occur, they look very similar to each other. So for instance, this is the phenotype you see high up in the Andes in Ecuador. And if you go to the lowlands, they look more like this. And this is because the birds learn to associate these Heliconius butterflies and their color patterns, particularly with impalatability. They're distasteful and toxic, and they notice quickly that they shouldn't have eaten this butterfly and learn to associate the color pattern with do not eat. And so it's beneficial for the species to look alike, because if the birds have already learned to recognize a color pattern as not to eat, it's beneficial to look like that as well. So they're all toxic. They're not trying to cheat the system like some other moths or butterflies. They are all toxic, but the signal still gets reinforced if they all look alike, and birds don't have to learn lots of different color patterns. And so we were looking at these two species in Ecuador. This is a data set mostly collected and lots of analysis done, particularly on the color patterns by Patricia Salazar and led by Chris Jiggins. So here in Ecuador, we have a hybrid zone from the lowlands to the highlands. And in the lowlands, the butterflies of erato look like this, and in the highlands like this. And melpomene looks very similar to erato in the lowlands like this and in highlands like this. And for me, very, very nice is that in between, in this hybrid zone, you find all kinds of combinations of these color patterns in both of the species. And particularly interesting is that in the middle, at mid-elevation, there is one particular combination of phenotypes that is particularly common. So at mid-elevation, we find this phenotype to be strongly overrepresented in erato and the exact same one in melpomene. So I wanted to know how these how this new phenotype came about and what's the genetic basis of it. So for that, we teamed up with Frank Chan and Marek Kuka from Tübingen University. And they developed haplotagging, which is a new link treat technology, where basically the DNA gets wrapped around beads, and then enzymes, the TN5 transposase heterodimers, they cut the DNA and add a short barcode, which is specific to the bead. So all the different fragments of this larger DNA fragment would get one barcode, and all the fragments from this one would get a different one. And then when you sequence those short fragments, you know from which larger fragment they came from, because they all have the same barcode. And that basically allows one to stitch together longer DNA fragments, and you get nicely phased data sets. And so we use this method on our Heliconius, erato, and melpomene data sets, where we sequenced 483 erato and 187 melpomene across this hybrid zone here in Ecuador. And then we looked at the beginning at, this is just a plot of FSD where we compare the highland versus the lowland butterflies. And you can see that there's very nice peaks. And what's particularly interesting is that erato and melpomene have the same regions with very high FSD, indicating that the same genes might be under divergent selection between the highland and the lowland butterflies. And because of these nice phenotype combinations in the hybrid zones, we were also able to figure out what those peaks actually encode. And so this peak here on chromosome 10 determines if you have the lowland haplotype at that region, the butterflies have 2 4-ring bands. And if they have the lowland haplotype at that region, they have just 1. And here on chromosome 18, this determines if the red is found in these parts of the wing or just in the 4-ring band. And so here we can really see which peaks are due to which, or encode for which color pattern differences, which allows us to really nicely link the phenotype and the genotypes. And this also allows us to have a look at this interesting new combination. And what we find is that this new combination, this new phenotype, which is at mid-elevation very, very common, it combines the highland haplotypes at 2 of those regions and the lowland haplotypes at 2 other regions. So basically, it's like a new combination of the genetic variants that encode these color pattern differences, which gives rise to this new hybrid subspecies or phenotype. And because of Müllerian mimicry, as both of these species basically have the same phenotype at very high frequency. So basically, it's like reshuffling Lego bricks again, only that now we know exactly what the Lego bricks are. And so also looking at other study systems, there's many people that when looking at their cases of rapid speciation, they find similar things. They find that admixture variation is very important for rapid speciation. Like for instance, in the Darwin's finches, there's genetic variants that encode the depth and shape of the beaks that have been repeatedly involved in speciation events, giving rise to this nice radiation of Darwin's finches. So we really think this is a general pattern where hybridization is involved in rapid speciation and rapid adaptation. But does hybridization really speed up speciation, or perhaps have we just now looked at cases with very rapid speciation, and does hybridization occur equally common in the slowly speciating lineages? This is a question I wanted to follow up because I was more and more thinking, well, we're just looking at these rapid speciation cases, and perhaps we're just thinking, well, hybridization is important in those, but perhaps the others hybridize just as much, and there it just doesn't seem to lead to diversification. So maybe this association, the evidence for this association isn't as strong as we think. So for that, again, I went to Ecuador, and now with my team of people, this is Eva van der Heijden, a PhD student, and Kim and Ismail are friends and collaborators in Ecuador. And with them and lots of others, we are now studying Ithomini butterflies. They also form Müllerian mimicry rings, so there's lots of species that mimic each other, and they're also toxic. Here another example of a mimicry ring, and many of the species you will find in both of them. And interestingly, also there's stratification, and we're higher up in the canopy. Some mimicry rings are more common than others. And what's also interesting in this group is that some of them are transparent. So this is 2 species of Ithomia. This one is not transparent, and this one is transparent. And this transparency seems to have evolved multiple times in different genera. And again, the birds learn to associate the different color patterns with impalatability. So they're very interesting in terms of speciation and ecology, but for me particularly interesting is that across the tribe of Ithomini butterflies, there's many lineages that have diversified very rapidly and others that have diversified at a much slower pace. Like for instance, if we zoom in here, Mechanitis and Mellinea butterflies have speciated rapidly, whereas their close relatives have not diversified, even though they exist in the same geographic areas. And so this provides me with very nice opportunities to contrast. Do we find that the rapid speciation genera are more commonly of hybrid origin than the slowly speciating ones? Or do we find any other factors that are associated with rapid speciation? And we are still working on this now with the team at the Sanger Institute, but in the Mechanitis butterflies that I just showed you before, which is a rapid speciation genus, we find lots of evidence for hybridization within the genus. We have not looked that much at hybrid origin yet, but we also find very nice cases of admixed species, like for instance, Mechanitis nesea, which together with our Brazilian collaborators, we found that they are partially of Lysimnia origin and partially of Polymnia origin. And they have lots of trait combinations that are intermediate between the two. And for instance, also this species has 18 to 19 chromosomes. This one has 14 to 16, and Nesea has 17 chromosomes. So in the next step, you're also looking at the chromosome evolution and how chromosomal differences facilitates reproductive isolation. And this is something that Joana Walker, the PhD student in my group, is working on. We're also looking at the genetic basis of the color patterns so that we can then study if those genes that are involved in speciation are more often exchanged through hybridization between distant lineages, perhaps, for instance, are involved in hybrid origins. And this is a project by Eva van der Heijden, also a PhD student in the group, where she found very nice peaks near cortex, which is a gene that has already shown up in the Heliconius work, and it determines here if the butterflies have a yellow band or not. And when we do CRISPR on them, we find that when you knock out this cortex gene, there's yellow stripes appearing on places of the wings where they shouldn't have yellow stripes. And so this phenotype is actually found in lots of different species pairs. And so we can then look if the genetic basis of this yellow band is the same one in these different species, and if hybridization facilitated the switch between the color patterns, which is needed, like if a species colonizes an area where there's a different color pattern that's at high frequency, a switch of color pattern is required, and if it can hybridize with the already pre-adapted species that have already the right color pattern, that might facilitate the rapid switch in color patterns. So that's something we're looking into here. And I already mentioned a bit, chromosomal rearrangements might be important in this group, and they might be extremely important because they are very extreme in this group. So most butterflies have actually 31 chromosomes, but these Domini butterflies range between 5 and 120 chromosomes, so they're absolutely crazy. And even within genera, like for instance, the Melinea species, which is one of the genera I mentioned as rapid speciating, they range between 13 and 30 chromosomes. And this is just known from colleagues from Brazil that counted basically the chromosomes. But now being at the Sanger Institute, we can generate beautiful chromosome level assemblies. And like that, we can actually see which chromosome fused or with which other chromosome and how are these chromosomes related with each other. And what we find is a huge mess. So basically, for instance, here chromosome 4 in Melinea marseus, in Melinea menophilus is chromosome 10, which is a bit fused with chromosome 9 in this species, which is here in chromosome 18, which is fused with 10 in this species, which is fused with 13, which is part of 6, which was split into 4 chromosomes. Yeah, so these are very, very closely related sister species. They're so closely related that mitochondrially they're identical. They can't be distinguished. But they show high reproductive isolation, and chromosomal evolution might play a key part in that. So together with Karin Neswal, who will start in April, and Patricio Salazar, who is already a postdoc in my team, we are going to look across this radiation to see if rapid chromosomal evolution or hybridization are associated with rapid speciation in these lineages. Yes, and so this is possible because I'm now at the Tree of Life program, which is basically an institute or a department within the Sanger Institute. We are 4 group leaders. Camille just started last October, and I started in July. And Mark Blackster and Mara Lanisak were basically the founders of the Tree of Life program. And one of the key projects of this program is the Darwin Tree of Life project, which aims to sequence all 60,000 eukaryotic species in Britain and Ireland. We have recently celebrated over 500 genomes complete. And yeah, so this is a huge sequencing endeavor, which means that there's a really, really nice pipeline already set up and different teams of people that take care of each of these steps, like one team dedicated to genome assembly, another one dedicated to genome curation, etc. And we can take advantage of this genome engine and study our butterflies and other study systems that I'm interested in. And with that, I would like to thank the collaborators that were involved in the Cichlid project. There's many others involved. I'm just highlighting the key ones here. And also of the different butterfly projects, particularly some key collaborators I haven't mentioned yet. And many thanks to my amazing team, and thanks to all of you for listening. And I'm very much looking forward to any questions you might have, or also comments or criticism.